convinced me that April and I needed to visit him at his retirement home in Gaeta, Italy. And he got me enthused about it. And well, we just picked up from there. Every year or so, he'd come back to the US for 3 weeks or more. And a cadre of us would meet at the Smithsonian and see where we were and figure out what needed to be done. And that's pretty much how I got into sciomyzids again. I had no passion for them when it was a job, but after it wasn't a job, I just found this amazing passion for them. So I think a lot of people find that. As I mentioned, I'm going to start with 7 minutes of a video that is a really great overview of the whole topic. And then I'll come back and we can get into some of the nitty-gritty. The only thing I want to ask is let me know if this isn't loud enough. I might be able to turn the volume up some. This is a 1971 video filmed by Shel Sandvid, the late Smithsonian photographer, narrated by Dr. Cliff Berg, who was the fellow who first noticed a sciomyzid larvae consuming a snail in a petri dish at his Alaska research stop. And by the end of that summer, he had reared out 6 more species. And what our textbooks up to then had called marsh flies were thereafter called snail-killing flies. So this is a Cornell 35 millimeter production from the 70s. So just bear with it here.
Most freshwater snails are scavengers. They feed on organic detritus and help to mineralize and recycle this material. However, some snails play an essential role in the spread of diseases caused by parasitic flatworms known as flukes. All fluke parasites of man and domestic animals must enter the bodies of snails and pass through some larval stages there before they can infect a new vertebrate host. Snails of the tropical genus Collaborator transmit a human blood fluke that causes a chronic, debilitating illness known as schistosomiasis or bilharziosis. It starts as an itching rash, followed by fevers, aches, recurring diarrhea or dysentery, increasing abdominal pain, and enlargement of the liver and spleen. The abdomen gets bloated, while the rest of the body becomes emaciated. Eggs of these parasitic worms leave a patient's body with body wastes. In water, they hatch into ciliated larvae, which survive only if they find a snail of a particular genus and enter its body. There they develop, reproduce, and transform into the second free-swimming larval stage. A few weeks after a ciliated larva has entered the snail, it begins to shed many thousands of narrow, fork-tailed larvae that are infective to man. These tiny larvae are barely visible without a lens. This view through a microscope shows their compact anterior ends and their forked tails. They are attracted to people who are swimming, wading, or just dipping their hands into water. They attach to the skin and digest minute holes through which they penetrate into the body. Then they enter a blood vessel, grow into adult worms or flukes, and a new case of schistosomiasis develops. This disease afflicts an estimated 200 million people and ranks among the most important health problems in tropical countries. There is no danger of catching it in cooler regions. It cannot be transmitted directly from one person to another, and the larvae of the first free-swimming stage cannot develop without penetrating into snails of the susceptible tropical species. All transmission of schistosomiasis could therefore be stopped wherever the snail hosts could be eliminated. Parasitologists have known this for many years, but chemicals used to kill snails are expensive to purchase and apply, only temporary in their effects, and usually toxic to fish and other organisms as well as to snails. Biological control by the introduction of natural enemies of snails may provide an inexpensive, longer-lasting control method with no danger of toxicity to other animals. Some strange-looking flies known as Sciomyzidae or marsh flies are being studied as possible snail control agents. No one knew until recently why most species are found only in wetland habitats. Then we found that the larvae are predatory on snails and will not accept any other food. Adult flies rest and mate on emergent vegetation and lay their whitish eggs in compact vertical rows on the stems of those plants. The newly hatched larvae enter the water, where they feed by killing small snails. They also forage out of the water on the wet soil of marshes and marshy shores. As they grow, they kill larger and larger snails. The fully grown larvae form floating pupal cases or puparia, from which adult flies emerge to complete the life cycle. Snails belonging to various species of Limnaea in different parts of the world are required for development of the liver-proof parasite of sheep, cattle, and man. Control of these host snails would curtail transmission of this parasite and limit the spread of the serious disease it causes. The tropical American snail, Biomphalaria glabrata, is easily killed by sciomyzid larvae. Other species of this genus that transmit schistosomiasis in Asia, Africa, and South America also are quite vulnerable to their attacks. The cloud of reddish pigment that will appear right beneath this snail's aperture shows how quickly a larva may rupture the blood sinuses and cause the snail to hemorrhage. Aquatic snail-killing sciomyzid larvae feed rapidly and leave their food snails as soon as their hunger is satisfied. When hungry again, they kill another snail, and each larva may kill as many as 24 snails. They never attack anything except snails, even when they are starving. Adult Sciomyzidae have a broad sponge-like proboscis, which they use for sucking up droplets of dew and flower nectar. They are not attracted to man or domestic animals and are rarely seen except in the marshes and swamps where their larvae are breeding. They also feed avidly on snails, both living and dead. Since they don't injure living snails, they probably just ingest the proteinaceous mucus that the snails secrete. Dead snails, which the larvae have killed but not completely eaten, probably make up an important part of their natural diet. These flies mate readily in laboratory breeding jars. In warm weather, entire life cycles of most species require only about 25 days. Adults commonly live 3 months or more in captivity. By laying a few eggs each day, each female may lay from about 100 to more than 500 eggs.
A voice from the past. So some of this talk will be technical, so take what you like and leave the rest. This is a little bit technical. It's about the taxonomy. The order Diptera, its flies, most of them have 2 membranous front wings. Some are apterous, they have no wings at all. The ones that you see the most commonly, housefly sized, are generally in the suborder Brachycera. Of course, you have mosquitoes and crane flies, those are in the Nematocera, but I have studied just Brachycera in my life. So how do you tell a Sciomyzid? First of all, size doesn't matter. I'll show you in a minute all the different size ranges. But the middle femur has a strong dorsal seta. I've circled it in red here. Another distinguishing feature is where the 2 top arrows are on that costa vein. Sometimes in some families there's a break. In Sciomyzidae there are no breaks at all. It's a complete costa, and the longer area points to a vein called the subcosta, and in Sciomyzidae, it continues intact all the way till it joins the costa. The third way to tell them, especially, for example, if you're looking at trays of unidentified specimens and you wonder if it's a sciomyzid, you see these two long setae or bristles. In Sciomyzidae, these are always parallel or divergent. They're never convergent or cruciate, which means crossed. Where are Sciomyzid found? Pretty much every region of the world except Antarctica. Clearly, they are more common in the northern hemisphere than the southern, with the Nearctic and the Palearctic having relatively equal numbers of species and genera. On this map, say, for example, North America, 201, that's species, 23 is genera. Get south of the equator and things taper off somewhat. You get over to the oceanic islands and there are only 4 species and 2 genera. The Oriental region is the least known. So there used to be 4 subfamilies. Now there are 3 because the Sciomyzidae of Australia and New Zealand, cladistic phylogenetic evidence showed that they were separated so far that they were elevated to family level. So now we're left with Sciomyzidae, which are distantly related to but are in the Sciomyzids. They don't parasitize snails, slugs, or clams. They are millipede parasitoids. Saltacellae are palearctic and afrotropical, which leaves us in the Western Hemisphere with just Sciomyzidae, and that's divided into 2 tribes. The Sciomyzidae, which are parasitoids, meaning one larva stays with one host, and the tribe Taxonomically, which are overt predators. They're the tigers of the marsh. They simply kill and eat as many as they need to reach the adult stage. In size, they vary from 14 millimeters on the big side, a species in Lesotho in South Africa, to Collaborator americana, which is found from Pennsylvania north. You people in Maryland just missed them. This is 1.7 to 1.8 millimeters long. Biochemical analysis, genetics, at the moment shows this arrangement where at the very bottom, the ancestor of the whole gang are those millipede parasitoids. The next line up, Saltacella, which is in the Palearctic and Afrotropics, is the next sister group to all the remaining groups. And then if you see the line with the 56, and above that the line 79, all of the genera in the group 56 are parasitoids in the Sciomyzidae, and all of them in the group 79 are the overt predators. And in the bottom 3/4 of that group, each of those genera is distantly related to the others. But when you get up toward the top, let's just call them the more recently evolved ones, you can see that some of these are more closely related than others. This is always open for change. When Lloyd and I were working together in the 80s, Cepodon and all the other Cepodon types, Cepodoninus, Cepodonella, Cepodonea, all those were thought to be the most primitive, but now it looks like they are actually most evolved. So what do Sciomyzids look like? I'm sticking to North America because it keeps this reasonable in length, this talk, but there are 23 genera known from North America, one of which is known only from Brownsville, Texas, which barely makes it North America. So there are 16, and I will only talk about 10 of them. People ask me, what do Sciomyzids look like? And if you look at these pictures, you can see it's really impossible to say, well, they look like this or that, because on visual characters, they're just so highly diverse. Sciomyzidae, the 6 genera on the left, Taxonomically, the 16 on the right, just to give you an idea of the fact that Sciomyzidae, there aren't as many different groups of them. Okay, so how do you tell Sciomyzidae from Taxonomically? On the front of this headless insect, I have two red circles circling the proepisternal setae, or bristles. All Sciomyzides except one species have those. No Tetanocerinae have those. This is the same specimen side view, and you can see that that is a fairly visible bristle. You don't have to worry about turning your microscope's power up or anything. If you see that, you know it's a parasitoid type of Sciomyzid. So now we'll go through 10 of these genera. This is Collaborator. These are the smallest Sciomyzids in the world. We have the smallest one here in eastern North America. The genus as a whole is found in the Nearctic, the Palearctic, and the Afrotropical regions. A paper that came out just over a year ago, 2020, by Bratt, Knudsen, Murphy, and Daniels, examines the biology, immature stages, and systematics of this genus, and it provides a complete overview of all aspects of the tribe Sciomyzidae. This paper took 50 years to publish, so having it in print is a very, very nice thing. This is rare in collections, primarily because the species are so small that most entomologists, unless they're collecting gnats, just leave them behind if they happen to have them in the nets. Furbelia has more species than any other in the Sciomyzidae, 96. Most of them look just kind of brownish. There's nothing particular about them. They're found in every region of the world and in nearly every moist habitat. Pteramicra is the same size or a little bigger than those tiny Collaborator, so they also are rare in collections. Most of the ones that I have found are in heavy shade in partly flooded woodlands, unlike most Sciomyzids, which you can collect in open swamps or wet sedge meadows. This genus is found in the Nearctic and Palearctic regions. A quick aside, a few years ago, in early March, I went driving around and I found probably a snowmelt pool, a couple 100 yards long and wide and about 4 feet deep. I went home and I got a gallon jug and I went back there with my hip waders and I filled a gallon jug with floating shells of a snail I later learned was called Planorbella trivalvis. They were just floating in the water and I thought I'd take them home and rear them out. So once I got back home, I took each shell and I held it up to the light source that I use with my microscope. And as you can see in the second photo from the top on the left, there's a pupa in that shell. All the ones with a pupa, I set aside in individual glass vials and I stoppered them with cotton wool and I moistened it every couple days to keep the thing humid. What I didn't know was if the pupae in those shells were from a summer from, say, 1920 or the previous summer. There just wasn't any way to know. But it was floating, so it was clear that the snail had been consumed. 15 flies emerged from these shells of 3 genera, including the one on the bottom left, which is Diptera trivittata. It was new for the state of Indiana. So this is a great way to get new species for different states. You don't find very many of these Sciomyzidae when you go collecting because most of them are only flying for a few weeks out of the year. And if you don't hit that, or if it's rainy during that time, next year maybe. But it's all kinds of fun to do things like this. So I'd encourage you to do some of this, get out and get them. Antiquita is a rare genus. The species are extremely specialized. Once they hatch out of the egg, the first instar larvae feed only on snail embryos in snail egg masses. This genus is found in the Nearctic and Palearctic regions. The second and third instars will attack small snails. My favorite genus, because they're so murderously difficult to work with and such a challenge, is enigmatic. These are often abundant, even in temporary roadside muddy drainage ditches. 3 of the specialists in the Nearctic are salt marsh snail specialists. Females of all 44 species and males of 43 of the 44 species require examination of the genitalia for confident identification. In other words, only one sex of one species can be told in the field. And although the genitalia of each of these is unique when you examine them under the microscope and you can identify them to species, people who have tried to make cladograms based on molecular genetics seem to end up with a clump of species or a clump of something not very well differentiated. So, does this mean that all putative species of Dictya are actually one highly variable species, or that this is a group that is just now evolving and partitioning up the resources? The evidence isn't convincing either way yet, but there's a paper of mine that's in review for publication right now describing 12 new species of Dictya. And I might say that George Foster has more specimens of one of those new species than anybody else in the galaxy, and he has made Columbia, Maryland, the epicenter of this new species. So, the distribution of Dictya is interesting. All but one are found in the Western Hemisphere, and that one species that is not in the Western Hemisphere is the only species in the Palearctic. One of our species actually dribbles down into northern Colombia, but that's it. So it's kind of our genus. And in the field, you can instantly identify Dictya because every one of them has a flat white face with a black mark right in the center. One of the most strikingly patterned and attractive species of Sciomyzidae is the genus Diptera. Right now, we consider there to be only one species in the US, and this is pictured here. It's Diptera flavescens, which up until a few of us published a paper a few years ago was called Diptera arcuata. But there was no reason for that change, so according to the zoological rules, the name goes back to flavescens. This species you generally find along the margin of woodlands or in the woods themselves. As evidence of this, in a 10-year trapping program in the forests of southeastern West Virginia, Pocahontas County, and adjacent Augusta County in Western Virginia, this one species outnumbered all the other sciomyzid species collected there combined. It almost certainly preys on terrestrial snails, but no one has yet reared them out to confirm that. This species is found in the Nearctic and the Palearctic regions. I've been working through the alcoholic material from that trapping program, and there are 3 discrete sizes of Diptera. So the feeling among people who have looked at them is there may be as many as 3 species involved. Time will tell. Limnia is a more colorful genus in the family, but it has some problems with it. As with Dictya, examination of the genitalia is required for confident identification of all but about 5 species, except unlike Dictya, which I have reared through multiple generations on our dining room table, thanks to my wife, no one has been able to rear any species of Limnia. The captured females will lay eggs, but the first instar larvae invariably starve to death. Researchers, including me, have offered them snails, slugs, clams, crushed flies, vegetable material, chicken eggs, larvae of other species of insects, sow bugs, spiders, and millipedes without succeeding in getting a single first instar larvae to live. Because of this, almost no female except in 5 species can be associated confidently with a male. So when you go to the museums and you look through the collections, most females are simply labeled Limnia species. This genus is found in the Nearctic and the Palearctic regions, with only 2 or 3 species outside the Nearctic. Seems like everybody's favorite sciomyzid is this little one. It could be called maybe the leopard of the swamp. Has a long name, Piscillagrapha decora. It's the only species in the genus, and it's only found in the Nearctic. As with Limnia, though, no one has able to get first instar larvae of this species to consume enough of any food to survive. Although, if you collect second and third instars, they will feed on snails, pupate, and mature adults will come out. In Indiana, I have found this spectacular species mostly around cold springs in leg-breaking deep holes concealed by tall hay in Amish fields. Why it is that what it is they prey upon in those highly special habitats, I have no idea. An aberrant genus is Rhinocera. The larvae of Rhinocera feed only on freshwater fingernail clams. The larvae can dive to the bottom of the bodies of standing water to find the prey. They can stay submerged longer than larvae of any other species of Sciomyzidae. 7 species of Rhinocera currently are known from the Nearctic and Palearctic regions. Rhinocera cresceni was described most recently by Murphy, Mathis, and Knudsen. That was 2018. And like a number of the people watching this, I suppose, as soon as the Smithsonian Institution reopens the National Insect Collectors to old fart researchers like Wayne Mathis and George, and I, we will be able to get into the collection, do a couple days of research, and complete our decades-long revision of Rhinocera. Sneak preview, it confirms only 6, not 7 valid species. And finally, Cepedon. This genus, with 79 species spread across all regions, is another very large genus of Sciomyzidae. The species shown here is Cepedon fuscipennis. It can be found in cattails across North America from the Atlantic almost to the Rockies. Where cattails grow, you just look above the waterline, maybe a foot or lower toward the waterline, and you'll find these cinnamon-colored, long-legged, sort of grasshopper-looking insects head down facing the water. I have heard that this is thought by college professors to be the Sciomyzid species in North America that is least frequently misidentified. So, the life history of Sciomyzids. The adults mate facing in the same direction. That's different in other families of flies. The male generally places its 4 tarsi on the inside margin of the female's eyes and uses its hind leg, as shown in the rightmost of those 3 pictures, to hold the female's wings open. Some males produce a droplet of some substance from their mouth, deposit it on the substrate, and the female comes over, and while she is imbibing it, he mates with her. The life histories vary between the parasitoid Sciomyzidae and the predator Tetanocerinae. The Sciomyzidae lay only a few eggs, many on the exact species of snail that the larvae will need, although some do just scatter them on the forest floor. And I say that because most Sciomyzidae are more forest or shade-loving than open marshland. The Tetanocerinae lays as many as 100, 200 on emergent vegetation like cattails. And when they mature, they come out of the egg and just drop directly into the water. I call them larvae. You might call them maggots. They emerge within 2 to 5 days. On the left in each of these 3 images is the business end that contain the mouth parts. The rest of the larvae is more or less like the skin of a sausage, getting ready to be stuffed with escargot. And then at the right end, you see those spiracles, which are the breathing tubes of the critters. The topmost illustration shows what are called creeping welts. These are found on larvae that are going to be moving around on mud. Larvae that are going to be inside a slug, a snail, or a clam, the bottom 2, don't have the creeping welts. So if you're into identifying Sciomyzid larvae, that's one quick way to tell if you're looking at a Tetanocerinae or a Sciomyzidae. So the parasitoid species can be aquatic or terrestrial. And as you might expect, if the egg is laid right on the host, it hatches on the host. And it usually develops within one single host. As they get bigger, though, sometimes they will abandon the first host if they've consumed it, find and kill a second host, and then pupate in that shell. The predatory species are almost all aquatic. They float on the surface. They don't dive except in the rhinocera. They swallow an air bubble as soon as they get to the water after hatching out of the egg. They can submerge at will to seek prey, but they don't go more than about an inch down into the water. And they move sort of like an accordion as they move around their habitat seeking these snails, but they don't have limbs, so they can't swim. Instead, they just jerk their head portions downward and then raise it slowly, and that enables them to move forward. Larvae around the world consume probably around 30 genera of snails. All of them are lung breathers. So I just picked out the 6 that are the most common here in the US. I was embarrassed when I was at the Illinois Natural History Survey, and I asked somebody over there what the difference was between right-handed and left-handed snails. It turns out it's the aperture. This one has the aperture on the right. You'll see some of the aperture on the left, but they don't have hands, so I was a little mystified. So anyway, Limnia, as you remember from the film strip, are common all through the tropics, and that's where Sciomyzidae has a reservoir. Then you have Oxyloma. These are land snails. Then Physa. These are back to the freshwater pond snails. And occasionally, late in the summer especially, if you have shallow water with a mud bottom, you can see up to 100 of these snails in 1 square meter, 1 square foot. Then you don't have a left or a right hand if you come out of an opening at the end of a ram's horn shaped shell, so this doesn't get to be right or left-handed. These ram horn snails include the ones that I found on that snowmelt puddle from which I raised the Diptera. And then Radix is another pond snail. And finally, Succinia or amber snails are terrestrial. Rhinocera, again, is specialized, and it only feeds on these fingernail clams. I don't know whose fingernails they were using to describe this thing, but I've never seen one anywhere near the size of my fingernails. They're maybe 3 to 5 millimeters across. If you want to try to find one of these, go along some stream that has had a high stream depth. What I'm trying to say is the water's receded after a high spring flood, and look under some of these leaves that have gotten pushed up, and you might see some of these fingernail clams. Not all snail-killing flies kill snails, although almost all species kill gastropods, snail, slugs, or clams, except for one species in Africa, there may be 2, that feed totally on oligochaete worms. So you have 3 species in North America that attack slugs as larvae, and the adults will scatter their eggs on foliage, vegetation, near where they expect the slugs to turn up. We have Tetanosteras plabegia, valida, and clara. Now these tetanosteras of all species, from what I've seen, produce a neurotoxin that stops the hearts beating in snails and slugs within a few minutes after those mouth hooks on the larvae bite the prey. I think somebody's actually working on finding out what this neurotoxin is, because even if a larvae attacks a much, much larger snail and it can't overpower it, and the snail produces copious amounts of mucus, that snail dies. So it's definitely gotten some neurotoxin. So how do these larvae attack the slug? Well, Tetanosteras clara larvae enter the slug right through the mouth and hang in there with its spiracles on the outside, breathing air, eating slowly until it has enough food resources to go through the second pupal state. When it hits the third instar, I said pupal state, I meant instar, larval state. As with a lot of these, when it hits the third instar, the last instar, it will devour everything, killing the prey. In some cases, the snail or the slug will stay alive right up until the third larvae, but then the larvae go for the gut lining, and that's it for the host. The other two species feed on non-essential tissues like Claire does for 10 to 20 days, a long time comparatively, before attacking the wall of the gut and killing the slug. So Tetanossera plebeiga enter the slug, not through the mouth, but by crawling under the mantle that covers the anterodorsal third of a slug's body. And you can see that best on the rightmost photograph. That raised shiny bit there is the mantle. So the larvae will crawl around on the slug until it finds the margin between the body and the mantle, and it'll crawl in there and eat while it keeps its spiracles outside so it can breathe. The larvae of Tetanossera valida enter the slug by crawling down and consuming the eye tentacles. That's something my kids love to hear. All three species are found throughout much of North America, particularly east of the Mississippi River. And I can generalize to say that the farther north you go, the more species there are, up to, say, the tree line in Canada and Alaska. So how does a sciomyzid larva consume its prey? You saw larvae attacking snails in the video. First, I just wanted to say that when an aquatic larvae attacks a snail, they don't both fall to the bottom. The larvae has swallowed air, it's buoyant, and also this is a close-up of the spiracular hairs on the back end of an aquatic sciomyzid larvae. They're hydrofuge, they repel water, and if you pull one of these larvae underwater and let go, it will pop up to the surface, and all the water will just run off of these hairs, leaving the spiracles available for transpiration and to prevent drowning. The business end, as I mentioned, is up front. You have three sclerotized pieces. You have the part with the mouth hook and the teeth and a transverse arch. Then you have a midsection, and then you have the main skeleton toward the back. And that skeleton is of major importance when you're trying to identify larvae, because you can measure the length of that long top margin and then the length of the part that goes back that's incised to the left and take a ratio. That, along with the number of teeth on the transverse sclera, gives you an index, and you can use that to figure out which it is. This is another example of the cephalopharyngeal skeleton. Here on the left are the mandibular sclerites showing the teeth, which on the top picture, it can be up to a dozen teeth, and the bottom, none. But lower down, you have secondary outgrowths that you could call teeth that range from none to 4. And then the ventral arches, the number of teeth in the ventral arch is specific to the species, although there is some overlap. This is the last thing some snails get to see. This is the close-up of the front end of an aquatic sciomyzid larvae. You can see the transverse arch. It's on the ventral side of the head, the belly side of the head. Above that is the oral cavity. And above that, those things that look like cross eyes are actually where the mandibular sclerites emerge. The eyes are actually at the very top. You see a couple of ocelli, or single facets of an eye. The larvae might be able to tell light from dark, but they probably can't form an image. As far as that nasty collar of hooks around the outside, imagine that you're one of these larvae and you've just sunk your hooks into a mucilaginous glob of snail flesh. It's going to be trying to retract into the shell, and what you've got around your neck is you pull away, it's going to grab onto that flesh and pull the muscle mass of the foot of that snail into the oral cavity. And then you just work your way forward until you've got a mouthful, and that's how it feeds. So the larval stages of both Sciomyzidae and Tetanosserae are similar, except that one is free-ranging and the other one stays in the shell of a host. They both consume non-essential tissues for a while, and then they consume the entire host. Some of these species actually will consume the rotting liquid black smelly remains of the host after it's dead. And a few species seem to wait until other larvae have killed and eaten most of the host and left it, and they'll go back and consume, well, detritus isn't the best word, but the liquid remains, the liquefied remains. And the chief species that I know that does that is one called Atrichomelina pubera. It's the only species in its genus. In the predatory species, the main difference is that they actively search out prey and they kill and consume 20 to 30 prey items. Now, if you are looking for biological control, looking for a tiger of the marsh that's going to get rid of the intermediate host of these infective organisms, do you want to work with a species that'll kill 20 or 30, or do you want to work with one that just kills one? That's why Tetanosterini are the biocontrol species that I work with the most. So a word about the pupae. The pupae of the Sciomyzidae are formed inside the shell of the host, and they take on the shape of whatever whorl of, say, the snail shell they wind up in. And as far as the free-floating Tetanosterini, they have a shape where the back end is higher because they're floating. They look like a brown grain of rice, and they want to keep the spiracles out of the water. This is the most frequent stage in which they overwinter. And by overwintering, I'm talking about the tropics. I mean, not the tropics, the temperate zones, North and South America. Although in the tropics, the wet and the dry season dictate when the breeding season is. The adult stage is actually probably the least interesting from a biological control standpoint. They basically are there to keep the species going. They feed on nectar, and as the film said, they will use their sponging mouth parts on the proteinaceous exudates of living or dead snails. When I raise these in the laboratory, I mix honey with powdered dry milk and a little brewer's yeast, and put a little bit of moisture on a bit of cotton wool, and that's all it takes to raise these. I'll put a popsicle stick in the vial, a broken piece for them to rest on, or a broken piece of cattail or sedge, and that's it. They just will lay their eggs wherever they feel like it. I take the eggs, put them in a petri dish with just enough water to cover a sand bottom, and the larvae are fed little snails that I can raise in an aquarium. It's very easy to do, except with Dictera and Limnia. So I've been retired since 2007. I don't live anywhere near the Smithsonian. So what do people like George Foster and I do? We work out of our homes. And April has been kind enough for the last 20 years to let me work in places like on our dining room table. This is what you look for in a partner. And I have to thank George for this beautiful microscope that I got last summer. I'd been using one for years and years that was okay, but this allows me to take pictures, videos, without driving all the way to Notre Dame in South Bend, where I used to have to drive about 2 hours. It was hardly ever even up to 0 degrees when I was up there, so this is a real boon. On the right hand is the way that I get specimens. Either I return from the Smithsonian with specimens that I found in the drawer after drawer of undetermined specimens, or in this case, I had one trip to Iowa this summer. Greg Courtney allowed me to be in the collection, the only person in the building for most of the day because of COVID and the fact that it was summer and whatever students were there in entomology were in the field. I brought back 1,000 undetermined specimens, and this is the way they arrive at Casa Murphy Sterling. The first thing I try to do is identify everything by external characteristics. The veins on the wings, the antennae, whether or not it has that proepisternal CT. But if I can't identify them that way, the next step is to look at the genitalia. And I made this video using that new microscope. It's not George Lucas, but you can take your forceps and you can see even on dried specimens, it's not attached very well to the body and it just can break off. Or especially on specimens that are pointed or with minutins, you can take a tiny pair of iris scissors and clip it off. What do you do with it next? Well, you carefully arrange the abdomens in these little spot dishes. Each one contains some distilled water and either lactic acid, a pellet of potassium or sodium hydroxide, and you leave it there overnight or longer. George Stacekel used to boil his, which is quick, but I found that it doesn't render photographable quality specimens. So I'm retired. I got time. I'll let these things just mature for a few days. The specimens from which these come are arranged in a tray in exactly the same order, because if you mess them up, you have no idea which is associated with which, and it's troublesome. So when I'm finished with this, I take each of these specimens and I put them in a little, about a 4 millimeter plastic vial, stoppered, and I put the pin with the specimen through that neoprene stopper, so it's permanently associated with the species, with the specimen from which it came. You already know why you study Sciomyzidae, but it's not just schistosomiasis. There are flukes all over the world that infect people through their feet, through their ankles, their legs, going through the hair follicles. And yes, you can cure these diseases with a pill. But if you're going back to that rice field barefoot the next morning and you're in a remote part of the world, that's not very useful. You won't have access to the pill. So that's why we continue to study sciomyzids for biocontrol. It works really, really well on islands. But when you're in a mainland situation, you have to keep reintroducing, because there are always incursions from existing populations. The example that started this whole biocontrol by sciomyzids was there was a giant liver fluke in Hawaii that prevented cows, sheep, horses, or goats from being raised. Within 6 weeks or so, these animals would get sick and die because they fed on giant liver fluke, which was in the grasses. So back in 58 and 59, scientists took about 20 maybe species of sciomyzids into the laboratory, and they exposed them to the one species of snail in Hawaii that was known to be the vector. And they found that Cipodomerus macropus loved that snail more than any others, and it really didn't seem to have any interest in other Hawaiian snails. So you have your silver bullet. So they introduced it, not because there were incursions from anywhere else as on the mainland, but because they had to build up a population. It was successful, but that word successful comes with, your air quotes. It got rid of the fluke. So they were able to clear off the native forests so they could have cattle ranches and horse ranches. I don't know that that's a success. Now the native Hawaiian limnid snails are under attack by these introduced species. So beware of those negative consequences. I'm going to skip this slide. We've seen the life cycle of schisto, and we know where it's found. The effects of schisto, it wasn't covered very well, but here are some visuals for the effects of schisto. These kids are holding up urine specimens. If it's red, that means they have schisto and they need to be treated. Schisto affects the old and the infirm, as well as young kids. People in the middle, they get anemia, lassitude, learning disabilities, but they will live to be older. And when you think about the fact that already in the Caribbean, it's in the Lesser and Greater Antilles. If you think of Cuba, for example, and you think of the geriatric makeup of places like Miami, it's not that big a jump to think that schisto could make it to Florida in the near future. So that's why we also continue to study these, make sure we know what we're dealing with species-wise, especially before it gets here. The Bahamas, too, is another one. Okay, I'm going to cover the history, and then I'm going to cover some current research, and that's that. The earliest specimen I've been able to find pictures of, or the actual specimen, was collected by a fellow named Charles Darwin, and this was about 1880. I never know how to pronounce this city anymore. In Indiana, we have Valparaiso, I've heard, but to be totally correct, I'll pronounce it Valparaiso. So, Enderlin, Hendel, Linnaeus, and Sack described a number of sciomyzid species, but they were described back then either as Sciomyza or as Tetanoceridae. In the 1920s, coincidentally, Crescent and Melander both produced what would have been, for each of them, for North America, the first really good coverage of Sciomyzidae. And they found out that they were both doing it at the same time, and they got together and made some agreements, but there are still some differences between the two coverages. Then came along George Stacefill. I'm going to go back one slide. And he was phenomenal. My office was next door to his for 10 years. He was a show and tell guy. Every morning he would come in with all kinds of really interesting stuff to show us. George, he described a ton of species, most of which are still valid species. And by that I mean some people describe 1,000 species, and 20 years later, 900 of them will have been found to have already been described. So the 900 are synonyms. But George was pretty darn good with his observations, especially considering that if you tried to look through his microscope, you'd be looking through crumbs of whatever he'd eaten during the last couple days. I was always amazed that he could see the detail so well using back then what we thought were pretty good microscopes. Murphy, quit hitting that button. 1960 to 2000 was the golden age of Sciomyzidae. Cliff Berg was the fellow who discovered that larvae consuming the snail in Alaska. He came back to Cornell, got massive amounts of grants from the World Health Organization, from National Institutes of Health, public health organizations, and he just happened to have a cadre of brilliant young researchers. Among them were J. Abercrombie, who became the South American specialist, Ben Foote, who reared species after species and described all the life stages, Lloyd Knudsen, who organized all the rest of us and kept us in the saddle and going at top speed, Rudolf Rogozhny and Jean Verbeck, who did the same thing over in Europe, Jean-Claude Villat, who is still active. He lives in Avignon, and he and I are working on some papers right now. Carl Valley was Mr. Diptera. He retired not that long ago from Pennsylvania Department of Agriculture and is helping a lot of us still. George Foster and I consult him frequently. And Jan Zuska. Jan was a great entomologist who, in a way, sadly, discovered that he had a supreme talent for painting, and that's what he did for the rest of his life. He has his own art shows and things. And for those of you who remember Ollie Flint at the Smithsonian, he was in that same cadre, but he got distracted and ended up working on Trichoptera, which are caddisflies. So who does that leave us? There are about 80 people in the world working on sciomyzids now and then. There are some Chinese who have started, some Koreans, and some have kind of fallen off. But Eric Chapman does the DNA work. He's at the University of Lexington. George Foster, you all know and love. He's watching this right now. Luciani Marinoni is down in Curitiba, Brazil. Rory McDonald did a lot of work in Ireland on using sciomyzids to determine the quality of the ecology of areas that are flooded in the winter and are cattle grazing areas in the summer. He got a job permanently in Oregon. That's where he's stationed now. Wayne Mathis, a lot of you know, he was head of the Diptera unit at the Smithsonian, and he is still out there helping lots of people work. We have some papers in the works. Jonas Mortelmans is a young man from Ghent, Belgium, who just popped out of nowhere a few years ago, an astonishing entomologist. He's sort of the future of sciomyzidology, I think. There's me, Jean-Claude Villat in Avignon, France, Carl Valley. Chris Williams was another member of the group in England and Ireland. He is in Liverpool, England, and many others. So that's where we stand now. This is Lloyd Knudsen and George Staceco in England in 1964. George was a character. You could look at his tie and know every meal he'd eaten the previous week. He changed his tie once a week. And although he had some eccentricity, he never got beyond high school. He taught himself Latin, Greek, French, German, all the Latin derivative language, the Romance languages. Super generous. So who cares if he changed his tie more than once a week? This is Ben Foote on the left. J. Abercrombie and Ben worked together for a long time. They lived about 10 miles apart in eastern Ohio. And Lloyd Knudsen. Carl Valley, Cliff Berg, and Lloyd Knudsen. When I started working at USDA, I had an office in Beltsville under the clock tower, but I also had to take a van downtown to do Lloyd's curatorial work because Lloyd Knudsen by then was the chairman of the Insect Identification and Beneficial Insect Introduction Institute. So the people that I was privileged enough to hang out with, which I did not appreciate back then, but boy. So in the front row is Ray Gagne. He still goes into the Smithsonian. His group is Sciomyzidae. And I used to complain that the genitalia of my Sciomyzidae were so small. And he showed me that the entire specimens he had to work with, for the most part, were smaller than the genitalia that I was working with. So I didn't complain after that. In the second row, it's Chris Thompson, the sadly recently late Chris Thompson, who was a syrphid specialist as well as a computer whiz. And then Lloyd Knudsen, Bill Worth, who studied simulids, ceratopagonids, and I used to joke with him, anything that caused human discomfort like biting. And George Staceco, Dick Foote, who was a Mr. Fruit Fly before Al Norbaum took over his responsibilities in that area. Dick Foote was a great wood carver and would carve belt buckles for all of us out of various native wood. And then Kurt Zabrowski, who had a responsibility for probably 50 families. And he was the only person I've ever met who apparently had memorized the Code of Zoological Nomenclature. And he loved that. It was kind of like memorizing a law book. The other people I did not know because they were in the mosquito world, Walter Reed's group, and they were in a different part of the Smithsonian. So a couple of quick pictures. This is Lloyd Knudsen, Jay Abercrombie, and me. Jay worked out at Fort Meade for a number of years, and my former co-student at the University of Maryland, Ben Pagic, still works out there. This is Lloyd and Jean-Claude in Avignon. Jean-Claude speaks some English, but I use his partner, Martine, as my in-between when we are together. This is Wayne Mathis and Jonas Mortelmans with something that came along last year that is seeming to distract him from flies. Takes a lot of energy to raise one of those. So as far as research, in the last decade, Lloyd published the book, Lloyd and Jean-Claude. It's 500 pages long. It's full of statistics and tables and charts, every mathematical thing you'd ever want to know about energetics. If you want to study sciomyzids, this is an essential resource. Shortly after that, some of us published what normally would be a catalog showing what species are valid, the history of names by which it was called, and what publication had published the most of them. By the way, I'm close to wrapping this up. This one has more than that. It tells where the types are kept, references to publications on the best distribution maps, if the larvae have been illustrated, if molecular analysis has been run. So it's more than just a catalog. And then in 2012, we got together, a bunch of us, we published a paper that basically said, where are we, what do we know, and what do we need to look at from here on out? And this being published in the annual review of entomology is like winning an Olympic event. And most people went out and had a drink that night, but I celebrated by getting proton beam treatment for prostate cancer. Yoo-hoo, wahoo. Okay, then this is something that those of you who study Diptera in Delmarva might know about. Wayne and Lloyd and I published this in 2008. It covered the 67 species in 16 genera recorded for Delmarva and 24 species in 10 genera from surrounding states that hadn't been recorded there. Then 3 Canadians and I published this online key to the genera of Sciomyzidae of North America. Actually, it goes to species, didn't change that in the title. This is online, and it covers almost all the species in your area. So I can give you the DOI, or if you want to see that, just look up key genera Sciomyzidae, and it should come up. Back in November, this finally came out. This is 12 years of research by yours truly. During this study, I visited all 92 counties in Indiana. I personally collected more than 10,000 specimens, and I found another 645 more Sciomyzids in collections around the country. And we now know that Indiana has 61 species and 15 genera, compared to Delmarva's 67 species and 16 genera. That's pretty darn close, considering that we don't have your high elevation mountains in Western Maryland or your salt marshes. These are what the distribution maps look like. You can tell most of them are pretty general, but if you look at map C, now there's a species that reaches its southern limit of distribution halfway through Indiana. And a lot of these maps will give you information like that. There's a website out there called sciomyzidae.info, where I occasionally update a new version of the bibliography of Sciomyzidae, a directory of Sciomyzid researchers, and as I go through keys and find ways to improve them or write new ones, those are up there too. It also lists obituaries and things like that. We thought it would be widely used, but so far, not so. And there's my email address. Anytime you would like to contact me, I love identifying Sciomyzids. And I would like to throw out some thanks. First of all, thank all of you for letting me speak to you on a subject that is near and dear to my heart. Again, Don Messersmith was the fellow who got me interested in this. And George Foster turned me on to the Maryland Entomological Society, which I'd heard about, but thank you, George. And I'll turn it back over to whoever's at the other end of this cyberspace.
All right. So thank you very much. That was quite a spectacular presentation. I really enjoyed the pictures. I'm sure everybody did. You have some really nice things you shared with us and a ton of information. And I always come away from a meeting like this realizing how little I know and how much more I could possibly even try to learn. So we can take some questions. Please, people, whoever would like to ask a question, turn on your mic and speak up, or if you want to put your hand up, signal. I see a hand. Warren. Yeah, Warren, go unmute yourself and
Thank you much. Yeah, great job, Bill. I have a little comment and story about Lloyd. As you know, I went to Madagascar a couple of times back in the 80s for Smithsonian research collaboration and collecting anything I could find. And I got 2 letters from people from different countries. One was from my parents, and one was from Lloyd Knudsen from Rome. and I see the label on the specimen on the letter, and who the hell is this? And I opened it up, and Lloyd is asking me, make sure to collect Sciomyzidae in Madagascar. And I tried. I did get my feet wet in a rice paddy, and I got a few, but nothing new, I don't think. But anyway, it's a really cool group to follow, and I don't know. I have lots more stories. Lloyd was instrumental in getting me some money through University of Maryland back in the 80s, working on those tiny little brown beetles, the Phalacridae, which I'm still sort of semi-known for. Yeah, he mixed a good Manhattan too. So
That was his drink. Warren, I don't remember exactly what year this was, but your phalacrids, the first time I met you, I was out in the field collecting on goldenrods, and I came into your office with that sloping roof. It was the dormer of an attic, and
yeah. Oh, I think.
Yeah, and I had a vial with a phylacrid. I didn't know what it was, but somebody had said, Warren knows everything. I was a good 15 feet from you. I held up that vial. You said, a phylacrid. I don't think many people could have told there was anything in the vial, let alone identify it. Yeah,
Little hemispherical,
yeah.
like bud scales. I mean, they're a very difficult group to study.
Yeah.
But that's another group that you have to pop the whole abdomen off and clear it to study the innards. And yeah, it's a tedious group. Anyway, good job.
Thank you.
Somebody had posted a question early in the talk about the possibility of the sciomyzids to control introduced snails in Florida. And I wondered if you might be able to speak to that. I can't remember the attendee's name, unfortunately, but if you're there, whoever that was, you can ask the question, please, if you want to.
That was Hess Muse. Yeah. Well, I think I've expressed my thoughts on introduced species, so anything to get rid of one is, I'm for that. We have the giant apple snails that have moved into Indiana recently, and people ask me, I'll get back to your question in a minute, what sciomyzid would go after the snail? Okay, it's this big. I said a sciomyzid about yay big, but we don't have any like that.
It's called a limpkin.
Yes, or a snail kite. Yeah, so it depends on the size of the slug. Some people would jump on this and say, you need to find out its geographic origin and bring over some sciomyzids that prey on it in its native territory. But then if it's effective and eliminates the leopard slug, it's going to go after our native slugs probably. So you have to look 2 or 3 jumps beyond what the immediate problem is. I'd like to ask a question. Is Dr. Prabhu Ganinger still on? Yeah. I have a friend in India named Kumar Gorpati, and no one's heard from him for a couple of months, and we just wonder how he's doing. So that was my question.
He appears to still be on this program. I see his name.
Oh, yeah.
We've got a lot of folks tonight from afar and many names which I don't recognize. I'm very happy that everybody could join us for this presentation. It's really good. I was particularly interested in the control aspects of schistosomes that you mentioned before. And you also mentioned there's several types of flukes that could be due to the control of the host. I wondered if Has there been some work done on control of bilharzia in the Caribbean, in Puerto Rico, for example?
I don't know that. Most of the researchers I dealt with early on were at Johns Hopkins University, and they're all dead or retired. So I haven't kept up with the schisto. That's an entire discipline in itself.
Yes.
Yeah, I know it's not south, it's not in Grenada or south. And since I spent most of my non-US time in Trinidad, I didn't pay much attention to the islands north of there. We didn't go wading when we were in Guyana, Warren. Yeah, Orinoco outflow, you get schisto there. So.
Yep. Yeah, I collected my water beetles by blacklight in Madagascar. I wasn't going to wade and dip them.
No. Lloyd went to Lake Volta in Africa once with, I think, Jean-Claude and some other researchers, and they carried a squeeze bottle of 95% ethyl alcohol, vodka. And every time a drop of water splashed on their skin, they would immediately wash it off so that these schistosomes would not bore through the skin.
Are there that many of them that you have to be so worried like that?
It's like COVID. You don't know who's carrying it.
Yeah, you can't.
So, yeah. Early on, when I was working for Lloyd, Dr. Gaboob was visiting USDA. They had just built a structure called the Aswan High Dam. Remember that?
Yeah.
Buried some old things. Well, he was a doctor, and his interest was in snail-killing flies because that dam created indisputably the largest contiguous schistosome habitat on Earth with aquatic vegetation, and Egypt's schistosomiasis rate went off scale. And he was trying to figure out how they could keep their population healthy from schistosomiasis. So he actually ended up staying with us in my townhouse in Greenbelt for a while. Sad ending to the story, he got schistosomiasis and he died from bleeding through his esophagus. He was about 30, he was a real young guy, but it can be a deadly disease. A couple years ago, Lloyd had told me year after year, when we finish this Delmarva paper, I know I'm going to die. So he kept that thing. Oh, let me just add one more. So he actually had a stroke a few days after we finished it, and he died a month later. I don't know if that was predictive or not, but as a result, his wife is not an entomologist. She's a dear, sweet person. We love her to death, but Lloyd had around 12,000 specimens in his office that belonged all over the world. More than half belonged to the Smithsonian, and he always meant to fill out the loan forms when he left places, but you know how it goes. Oh, geez, my flight's coming. So I volunteered to go to Italy, and Jean-Claude Villat drove down from Avignon, and the two of us spent the better part of 2 weeks just trying to figure out whose specimen is this. And Jean-Claude took the specimens back to the museums in Europe, North Africa, and the eastern part of the Palearctic. And I took them back to the Smithsonian and prepared them, and they sent them back to the museums, except for Cornell. I had so many at Cornell's, I just drove them to Cornell and put them back in the collection. But word to the wise, if you're not partnered with somebody who's an entomologist and you have a lot of somebody else's specimens, think about labeling those real carefully about this box upon my demise needs to be returned to somebody. Don't leave that for your partner.
Good point. There's a message in the chat box from Dr. Prabhu Ganigar, who is inquiring about some work in India and ask if there's any help in identification for him. So if you look at the chat, Bill, you can see that. And I should remind that I think you posted your email in the chat already, didn't you?
No, I'll do that right now.
Oh, okay, yeah, that'd be wonderful if you could, for anybody interested in that. Any other questions while we do that? Anybody have something they want to ask or mention?
I've got a quick question. Bill, freshwater versus estuarine versus marine, how, do you have more species in one habitat versus other habitats?
I only know 4 species that can tolerate salt. 2 of them are mid-marsh. One of them is high marsh, if you know what I mean by that.
Yeah.
So I don't know any that can stand saline water. There's a kind of Sciomyzid, is it, that attacks barnacles? But that's the closest sciomyzid relative that does that. If you go out into a typical marsh on the Eastern Shore, well, George can address this better than me. You will probably find Haplodictia setosa and 2 or 3 species of Dictia, and that's it as far as sciomyzids go.
I was wondering, with a place like Hart Miller Island, which is in the upper bay, it's not very saline, and you get a lot of freshwater pools on the island itself.
You will get Limnia, Dictia, probably some Furbelia, Cepidon if you have tall enough vegetation. Yeah, I think the upper bay these days is 100% fresh water. And the expert on that is probably Mr. Crab Catcher Warren Steiner.
Well, I've done a lot of collecting in the Rehoboth Bay area over many years. I got a boat, and there's a one site that I can only get to by boat. And there's been 2 sciomyzids I've collected there. One of them is called Dictia oxybiles, and it's consistent. Every year I get 1 or 2 of them. They're not a lot, but it's only there. It's in the on the salt marsh, really close to the bay itself, close to the dry beach area. And of course, every now and then I get this Haplodictia. What is it, setosa, Bill? Yeah,
Yep.
Down there, yeah. Got like just a couple of them, but oxybiles seems to show up 1 or 2 here and there every season.
There were 5 species of Haplodictia. 1 west of the Rockies, 1 down south, 1 all through the east, and then another one that was found in Florida. There was a species known only from Bermuda. But Norm Woodley conducted a pretty comprehensive Diptera survey there a couple decades ago, and that species is now extinct. All the marshes are now condominiums, and there's no habitat left for that one.
And I'll put in a plug for the 2023 issue of the Maryland Entomologist, since we already have the 2022 issue full. But George Foster has submitted a paper on the sciomyzids of Howard County, Maryland. A very lengthy paper that gets into a lot of details about sciomyzids in the central part of Maryland. So you can look forward to that in the 2023 issue. If any of you are not members of MES and might be interested in it, I can put my email address in the chat too. It costs a whole $10 a year to join, and you do get a nice journal once a year.
That paper is due a lot to Murphy's efforts in editing that thing with me. So he has been a great help.
Very good.
Thank you, George.
Anybody else would like to ask anything?
Fred, are you Romanian?
Am I Romanian?
I'm looking at
No.
your name. No, like Kaczescu.
There's about 6 letters missing on it right now. There's
Oh, wait. Kedis. Greek?
Paraskevu, yeah, Greek, Paraskevoudakis, is the full name.
Okay.
I'm not sure what you're seeing on the screen there, but yeah, Paraskevoudakis. And when they end like that with Akis, it's from the island of Crete, in case you didn't know.
Oh,
Also a genus, of Tenebrionid. Akis.
Oh, is that right? A tenebrianid, really?
It's a pretty big Mediterranean flightless black thing, but Google it. Akis tenebrionidae.
Spell it again, Warren, I'm sorry.
Akis, yeah.
Oh, AKIS.
that's the whole name? AKIS?
That's the genus name. Capital A.
Warren, you were always the expert on tenebs.
They're,
man.
well, it's my group. I can't help it.
I know, you loved them.
Now, speaking of which,
I see why.
I have some from Uzbekistan that I want to show you.
Yeah, cool. Yeah.
We had
Are
a
you-
member, somebody joined us tonight whose name is Socrates Latana.
Yeah,
Wondering where you're from, Socrates. Are you still listening?
Yeah.
He's muted. I'm not sure if he's still there or not.
Well, I see Kevin Barber, and if this is the Kevin Barber from the Great Lakes Forestry Research Station in Sault Ste. Marie, we almost met twice. Once when I went up to his station as he was leaving, and once when I left when he was returning. But he gave us some great tips on where to get some sciomyzids that are restricted to the Western Great Lakes area.
Hmm.
Yep.
So if anybody's interested in more information about the Maryland Entomological Society or contacting about anything, I'm posting my email, and you can shoot me an email if you wish to. It's in the chat box now. So and without any further ado, if there aren't any more questions, of course, anybody wishes to stay and just chat a little is always welcome. But we don't want to keep Bill any longer. So if anybody who wants to leave or needs to go is all fine. And thank you, everybody, for coming and joining us tonight. And I hope that maybe we'll see some more folks in the near future. Our next speaker is going to be David Adamski, also a Smithsonian person on micro moths. Is a group that he's been working on for some time with another gentleman, and the name slips my mind right now, sorry, but we'll be posting that information in our upcoming flyer and newsletter. And we're going to try to do an in-person meeting at UMBC where we have met in the past, and kind of make it as a hybrid so that we can do a Zoom and the in-person meeting. And I'm not sure how technology is going to allow, but we're going to attempt that. And hopefully we can pull this off. And that way we can keep people coming from everywhere, because the advent of Zoom has made the world smaller, so to speak. So in any case,
Murphy, that was a great talk. I enjoyed every minute of it.
Yeah, can't wait to see you guys again. Hey, Fred, there were some entomologists in Europe who said that this talk was in the middle of the night for them, and they wondered if they could get the URL and watch it when it wasn't so inconvenient.
yeah, I got a few emails already, Facebook posts and whatnot,
Okay.
and I intend to make that available for them as well. One guy's name is Dan. You probably know him, Bill, I'm sure. can't think of his last name now, but
Oh, he
Dan
was-
Young.
Oh, yeah.
Sound familiar?
You know,
Dan Young.
Captain Dave.
He's where? Texas?
Wisconsin, I think.
Bill, I did record this.
Yeah, Wisconsin. You're right, Warren. He's University of Wisconsin.
Oh, okay.
Yeah,
He's another beetle guy.
professor of entomology and director of the University of Wisconsin Insect Research Collection.
Mhm.
So, yeah, but I'll be posting this for folks like him and whoever else. So,
Yeah, Bill, I'm asking you after the fact if it was okay to record this because I already recorded it at this point.
Well, I had emailed Fred asking him if it was possible to do that for Megan Crew and some of the others who
right.
were in a different time zone.
I have it
Thank you.
recorded on my computer. Now I just have to figure out what to do with it now that it's on my computer.
Oh, you could post it to Facebook.
Oh, the link.
There's a link, you put it on Facebook and then it can be retrieved easily from the Facebook.
Well, I'm one of the few that's not on Facebook, but we can work that out.
Did I say, wait, I meant to
either.
say, no, post it, I'm sorry. Did I say Facebook? Post it on Zoom, on the Zoom platform.
Okay.
And then it can be taken off a Zoom platform and as a link, so.
Well, I was a Zoom meeting virgin before tonight, so now I know where the share screen button is.
Fred, is it possible for someone to get make a link for it and send that link out to all the MES members? Unfortunately, I signed in a few minutes too late. I do
Yes,
want to
Mike.
get-
Yeah, I think we'll do that. We can do that.
Michael, that is a fantastic T-shirt.
Yes,
I love it.
yeah, nice.
I can wear a different T-shirt every day of the month.
I want to
And
show
they're all
off my
insect
new
related.
James Webb T-shirt.
Well, that's a nice
Any entomologists who are also amateur astronomers.
Oh, and I have my cicada T-shirt on
Nice,
That's
that's good too.
from last year.
That's a very nice one, actually. I don't have a T-shirt.
Oh, poor Fred.
But I can get one from Mike. All of you could for a mere $11. They're beautiful shirts. I have to say, giving you a plug, Mike.
Mhm.